Hi, my dear students. Welcome back to my channel once again. I hope all of you are doing so great. So, let's start today's session without any further delay. Today, we are going to discuss a very interesting topic that the the creation of the living cells in the lab. We have already covered the chapter the cell, the fundamental unit of life, right? You can go and check out that videos of the chapter. Okay, there are 4 lectures of the chapter. Cell is the basic structural and functional unit of life, and of course all the living organisms are made up of cell. We know that, right? So the human beings are also able to create the cell, that too a living cell in the lab. So let's check out that this research paper. So basically, we can see that the researcher named Ronit Freeman with her colleagues from University of North Carolina Chapel Hill, she published her research in the in Nature Chemistry. Okay, so Ronit Freeman and her colleagues, what they did, they made the living cell, which exactly look like and act like all the cells from our body. Okay, so isn't it very interesting that now human being is also, they can also create the living cells. Okay, now how they create it. So basically, they manipulate the DNA and the proteins. Okay, proteins are the building block of our life. We know that, right? So all the cells have the protein in their structure. So they manipulate it, the structure of the DNA, and then they create the cell. So first let's discuss that cells. What is the cell? As I already told you, cell is the structural and functional unit of life, and cell is basically made up of the proteins. Okay, we have already studied about the cell membrane, the structure of the cell membrane, where we can see that proteins are embedded into the cell membrane or plasma membrane. Okay, so that means the proteins are very much essential for forming the structure of the cell. Okay, and cytoskeleton also, in the cytoskeleton also, the microtubules are made up of proteins. Okay, which is the cytoskeleton is basically uh, uh, forming the shape of the cell, maintaining the shape of the cell and allowing the cell to be flexible. Okay, so we can see that proteins are very essential for the formation of the cell and for the functioning of the cell also, because we have already discussed in that chapter that ribosomes synthesize proteins and proteins are very much important. Okay, so now, how Ronit Freeman, what they did basically in their research. So they do not use any natural proteins. Okay, isn't it amazing? So how they made a cell? Because we know that proteins are essential for the cell. So without using any natural proteins, what they did, they built up a new programmable peptide DNA technology. Okay, new DNA technology. So basically, they program the DNA sequences, and that modified DNA sequences now it can direct the peptides, means that modified DNA sequence which is reprogrammed, it is directing, or you can say it is giving instructions itself to the peptides. Peptides are the basic unit of the proteins. Okay, so that reprogrammed DNA is giving instructions to the peptides for what? To arrange itself in such a way that it can bind together and make a cell. Alright, isn't it amazing? So, the reprogrammed DNA sequence are giving instruction to bind the peptides together in such a way that it can make a cell, and it exactly looks like the cell which are present in our body, and it is a living cell. Like in look wise and in function wise, every wise. Like it is a like duplicate, you can say. Okay, so now we can say that human being, we can also make a cell. All right. So now, what is the benefit or what is the difference between this original cell, original living cell that we have, and this synthetic living cell, we can say? Okay, what is the difference? So, the original living cell, which we already have, like we are made up of cells, so it is susceptible to the hostile environments and the severe temperatures also, right? We are very vulnerable to the severe temperatures. You can see in summers there is 47 degrees Celsius outside, and then like we are rushing towards the air conditioner room. है ना? We cannot, we are not able to tolerate that much temperature also, right? But this synthetic cell is stable even at 122 degree Fahrenheit, that is 50 degrees Celsius. It is stable in that Celsius also. Okay, so this is opening the possibility of manufacturing cells with some extraordinary capabilities. Okay, so that means in future we can make some cells, the living cells, which has some extreme capabilities, like it can survive in that extreme environmental conditions also, right? We don't know, in future there could be like due to global warming, temperature is already increasing. So in future there might be the like very high temperature, chances of high temperature, so that this cell can be able to survive in that temperature also. Okay, now currently where we can use this technology, what is the application of this technology? So basically, we can use it. This living cell, living synthetic cell, can be used to prepare the regenerative medicines, okay, the drugs delivery system, and some diagnostic tools also. Okay, so this living cell can go inside the body, it can do the diagnostics process, okay, without hurting the patient. All right, it can deliver the medicines inside the body also without even hurting the patients or damaging the patients, right? So it can be used in a medical science. Isn't it amazing, right? So, uh, yes, so that is about the preparation or creation of a synthetic cell in the lab, and that is a huge success in the field of science, we can say. So that is it for today's topic. Thank you very much, all of you. If you like the video and if you want more such videos regarding discoveries and the creations and the researches in the field of science, let me know in the comment box. I will bring more such videos to you. Thank you everyone, and please hit the like button and share and subscribe to my channel.